Hello everybody, welcome back to the My Tutor online school this afternoon. We have a GCSE Biology session on treating, preventing and curing disease. I'm really excited about this session actually because in last session we had lots of questions asking about how to treat, prevent and cure diseases. So hopefully it's really relevant stuff for you guys. If you're new to the My Tutor online school, welcome. This session will be an hour long and it will be recorded. So provided that you've signed up, then you'll be able to get a link to access this session afterwards. And just to say that all sessions are an hour and there's 45 minutes for the tutor to deliver the lesson content and then 15 minutes at the end for questions, but you can send questions through before that using the chat function or the Q&A function. And we have our tutor, Charlotte, delivering this biology session. And I think lots of you are familiar with Charlotte now because Charlotte has done all of the sessions before, but if you aren't familiar with Charlotte, then Charlotte has done lots and lots of lessons with my tutor and she has been leading all the biology sessions on the online school. And she also studied conservation and biodiversity at the University of Exeter. So I'm going to hand over to Charlotte now to lead the session for you all.
Perfect, thank you. So I'm going to move over to the My Tutor whiteboard now to start. So this session we're going to look at treating, curing and preventing disease. And last week we finished with a lesson about pathogens and the different sorts of pathogens that we could get. So we had some really nice pictures of lots of different diseases and we ran through whether those diseases were from bacterial pathogens, viral pathogens, protist pathogens or fungal pathogens. So we had a look at those 4 types of organism and the diseases that they cause. And we ran over what a pathogen is. So a pathogen is a microorganism that causes diseases. So pathogens can be fungi or bacterial or viruses or protists. On the other hand, if you've got bacteria aren't always pathogens, so they don't always cause disease. We do have things like good bacteria in our guts that don't cause disease but are really helpful. So I thought we'd start with using the Q&A function. I thought we could start with a question with what ways can you think of for treating, curing and preventing disease? So if you pull up the question and answer box and type in any ways that you can think of for treating, curing and preventing disease, we'll see what sort of answers come up. Okay, so we can see a couple coming into the chat. Ready? Good. There's loads here. We've got vaccinations leading to herd immunity. Someone's done some homework there. We're going to learn about vaccines today. Self-isolation, yes, very good. So we're at a time at the moment, obviously with coronavirus, that's why we're all at home, we're self-isolating to try and stop the spread of that disease. Lots of people saying vaccines, antibiotics, cleaner workspace, yes, for example in the kitchen as well, keeping the kitchen clean when you're preparing food, good. Chemotherapy we've got as well for treating cancer, yes. Stop mosquitoes mating or biting, either of those would work well for malaria, and we talked about how mosquitoes are a vector for malaria last time. Social distancing, very good. Fungicides we learnt about as well last time, so any sort of pesticide spray that you use in agriculture, or sometimes they're pellets as well, they can be used for some pathogens in plants. Antibiotics, good, we're going to learn about those today. Masks, radiotherapy for cancer as well. Make sure your immune system is strong. Don't go to the swimming pool. Someone did come last time when I said that verrucas and athlete's foot can be caught at swimming pools. That's true. Great, loads of good ideas here. Transplants, yes, we learnt about transplants as well when we talked about heart transplants. White blood cells, we're going to learn more about those today as well. This is great. Okay, so let's explore some of those ideas more then. So, before we go into vaccinations or antibiotics or even our immune system, we're going to talk about what on the outside of our body helps us to defend against disease. So we talked about this briefly last time a little bit right at the end, but we need to go into it a bit more detail according to what's on the specification. So our first line of defense against disease is the outside of our bodies. And we have several physical barriers against infection. So I'm going to underline physical there in red. So first of all, we've got our skin, covers almost all of our body, and if cut or grazed, it heals fast, forms a scab. Our skin, if you have a look at your skin, it's pretty much impermeable. It is a really good barrier for anything trying to get into our bodies. In our nose, we have internal nose hairs. They're actually really important, even though sometimes they don't look very nice. So cells in the nose produce mucus which traps pathogens before they enter the lungs. So those hairs in the nose help trap some of those pathogens in the mucus. And then when your nose is blown, the mucus and the trapped pathogens are removed. So that's why when we have a cold, we blow our nose a lot. That's our body trying to get rid of the pathogen. And we have kind of a similar situation in our trachea and bronchi. So our trachea is the pipe that goes from our mouth down towards our lungs, and then the bronchi are the pipes that take the air across our lungs. Cells that line the trachea have cilia, which are tiny hairs. You can see them on the diagram here. So these cells that have hairs are called ciliated cells. And then we also have these goblet cells here, which make mucus. So between the two of them, so the goblet cells make the mucus, and then the ciliated cells use their hairs to waft the pathogen and mucus up towards the throat, so away from our airways, up towards the throat to be swallowed. Then the pathogen goes down to the stomach. So then we move on to our chemical barriers. So some parts of the body don't have skin. So for example, on our eyes, and on our eyes, we have tears. And tears contain enzymes that help prevent infection. So the same as our saliva as well. So we can see from this diagram, they've got antibacterial enzymes. So that allows them to stop pathogens using those enzymes to destroy them. Also got our stomach. So we've talked about the skin and about mucus. So in our stomach, we have good gut bacteria, which outcompete the bad bacteria usually. So we've got kind of a natural selection of good bacteria within our guts. But our stomach is full of acid. So it has a very low pH. It's got lots of hydrochloric acid. That's the type of acid there. And it's a strong acid that kills pathogens that have been caught in mucus in the airways and then swallowed down to the stomach. So the really low pH just kills those harmful microbes. So that's our first lines of defense. Then we get to our second line of defense, which is our immune system. A couple of you have already talked about the immune system and how keeping our immune system healthy helps us fight off pathogens, which is very true. The key parts of our immune system that you need to know about are our white blood cells. White blood cells can protect us from disease in 3 ways. So to start with, we talked about this briefly before when we talked about blood, so this might not be new. But phagocytes are the first type. They are white blood cells that engulf and destroy pathogens. So they send 2 arms around them, they bring them into themselves, and then enzymes within the white blood cell digest the pathogen. And this is called phagocytosis. That's a key word there. Okay, then we have lymphocytes. Lymphocytes are white blood cells that produce antibodies. So lymphocytes are white blood cells that recognize proteins called antigens that are on the pathogen. So they recognize those antigen proteins and go, well, this one shouldn't be here. That bacteria shouldn't be here. What they can then do is produce antibodies, which are the same shape as those antigens. Those antibodies then bind to the pathogens, and they make the pathogens kind of stick together. So they make it really sticky, which means that the phagocytes can come along and eat loads at once, or engulf loads at once, in phagocytosis. So as well as antigens, white blood cells can also produce antitoxins. So some microorganisms produce toxins, and our white blood cells can detect that, produce antitoxins, and then the same principle applies. They can kind of neutralize that microorganism or that pathogen. Okay, so we had some feedback last week saying that mini quizzes would be quite handy part of the way through. So if you go to your chat functions again, and we'll quickly do these 3 questions. So question 1, what kind of barrier is your skin to pathogens? Is it A, physical, or B, chemical? Good, lots of A's coming through there, perfect. Okay. Brilliant. Question 2, what kind of cells produce mucus in our airways? Try that one. Yeah, lots of B's coming through there, brilliant. Goblet cells, not goblin cells and not ciliated cells, goblet cells. And question 3, what type of white blood cell engulfs and destroys bacteria? Is it A, lymphocytes, or B, phagocytes? That one should be B. So if we flip back to the slide before, so it's the phagocytes, this top line here, that engulf and destroy in phagocytosis, and it's our lymphocytes, that word has been cut short a bit there when I've put the slide up, that produce the antibodies. Okay, any questions? Pop back slide 3 there for someone. Any questions so far? If you type any questions that you've got now, as we've got quite a lot of things on the chat. So you might, if you've asked some before, you might need to retype. Can hydrochloric acid kill COVID-19? Honest answer to that is I don't know. I'm not sure what role our stomachs play. So COVID-19 affects our lungs. So in theory, yes, because our bodies would be trying to get that pathogen into our stomachs, but I'm not sure. I don't know. How can aplastic anemia affect you? Ooh. Is the first layer of our skin dead? We do have a lot of dead skin cells. I'm not sure that all of the first layer is dead, but we do shed the first layer of our skin regularly. How can aplastic anemia affect you? No. I'm not entirely sure about aplastic anemia. I'd need to read up on that one. And is this content for all exam birds? That's a good point. We should have gone through that at the start. So this content is tailored particularly for AQA, but what we've tried to do is to make sure that it covers the topics that are in all other exam boards as well. So you may find that when you go back to school, some of the examples and things are a bit different, but the topics should be broadly similar. Okay, let's move on to the next section. So next we're going to talk about vaccinations. So you need to know how a vaccine works, and you need to be able to talk through this graph. You'll get lots of questions, lots of past exam questions about this graph. So here are the stages. There's diagrams from BBC Bitesize. They do a really good vaccination section if you're looking for good revision tools. So the syringe injects an altered form of the pathogen. They normally inject a weakened form of the pathogen and a small amount of it into the body. This then means that our white blood cells, they encounter that pathogen, they recognize those antigen proteins that we talk about and go, hey, this one shouldn't be here. And then they release those complementary antibodies. So this is our lymphocytes that we talked about, release the complementary antibodies. They attach and clump the pathogens together. And then our phagocytes come along and phagocytosis occurs. So they engulf and destroy the pathogen. So basically, our vaccination triggers our immune response. It triggers our white blood cells to make those complementary antibodies. This means that, so our bodies are very clever. If we then talk through this graph, so first exposure to the antigen is when vaccination occurs. This point here is where we were vaccinated, and the immune system then produced these antibodies. So you see we've got antibody concentration on the y-axis there. So it's taken, say, a couple of days to produce that many antibodies. They fought the pathogen, and then we don't need as many antibodies, so the concentration's gone down a bit. Then, if you go to school or to anywhere else, public place, and you happen to be exposed to that antigen for the second time, so that same pathogen again, but this time it's not a vaccination, this is the full form of the disease, your antibody concentration increases rapidly. So you produce a lot of antibodies very fast. And that means that, so your immune response is a lot quicker, and then it gradually slows down as it combats the disease. So this is the key part of vaccination. Vaccination means that if we do ever catch the disease, our immune system remembers it and it responds quickly and it gets us well quickly. So the disease, the pathogen doesn't have time to act too much before our immune system has got on top of it, so we don't show as many symptoms. So for some diseases where we don't have a vaccine, if you can get the disease once, then again, your immune system will recognize it and then it will get rid of it again. So that's why you tend to only get one sort of cold once, or chicken pox is the classic example. We get it when we're at primary school usually, and then that means that you never get it again. How does vaccination help prevent disease? Herd immunity. So this is the next thing that you need to learn. So vaccination in one person has the effect that we talked about before. It means that there's a rapid immune response the second time you're exposed to the disease or the pathogen. So how many people are vaccinated makes a big difference to how much of the population becomes infected with the pathogen and gets the disease. So we've got three scenarios here. We've got blue people who are not vaccinated, but they're still healthy. We've got people who are vaccinated and healthy, they're the green ones. And then we've got orange people who are not vaccinated, they're sick and they're contagious. So with hardly anyone vaccinated, so we've got lots of people there that are not vaccinated but healthy, and we've got a couple of people that are not vaccinated and have got the disease. The contagious disease spreads through the population. That's what we've got at the moment with COVID-19. We don't have a vaccination for this disease, so we're all susceptible and we could have mass infection through the population. What we're doing about it is trying to keep ourselves separate, because if all of these people in this square weren't standing so close to each other, they wouldn't be exposed to the pathogen so much, so less people would be infected. In the second scenario here, we've got some people who are vaccinated and healthy. And so the contagious disease only spreads through the people that are unvaccinated. So you've got more healthy people in the second scenario, but you've still got quite a lot of people getting the disease. However, if you vaccinate a majority of the population, like in this third scenario, lots and lots of green people here, that means that the spread is contained. So without us all being 2 meters apart, we've got enough people who are vaccinated acting as barriers that they can protect some of those not vaccinated but healthy people from not vaccinated, sick and contagious. There's just less chance of them encountering each other. And bear in mind that it's not possible for everyone to be vaccinated. Some people have other health conditions that prevent them from being vaccinated from a disease. So the idea of having the majority of a population vaccinated is that they can protect the ones who can't. There's lots and lots of very successful examples of vaccination and how that's worked. So a lot of the diseases, we talked about measles last week. We're all vaccinated in the UK for measles at a young age with the MMR vaccination, which stands for measles, mumps and rubella. I'll write that down because I know some people wanted more information about that last time. Measles, mumps and rubella. We've had, I think, a 97% reduction in measles, mumps and rubella in the population since vaccination was introduced in, I think, the 1950s. So vaccination works incredibly well. And it'll be once we have a vaccine for COVID-19, that's what scientists are working on now, that will be the most effective way of controlling the coronavirus. Okay, mini quiz. So head to the chat. 1. Vaccination injects what form of the disease? A. An altered, weakened form, or B. Large amounts of a strong pathogen? Good. Lots of A's coming through. That's what we need. Fantastic. Okay, question 2. Vaccination ensures what sort of response to a second infection by the pathogen? A. Slow, or B. Rapid? Good, lots of B's. Fantastic. And then question 3, herd immunity means that most people are A, vaccinated and healthy, B, not vaccinated but still healthy, or C, unvaccinated and infected. A, good. That's exactly what we need. So herd immunity means that most people are vaccinated and healthy. Okay. Any questions about vaccination? If a vaccine is a weakened or altered version of the disease given to prevent getting the disease, then how come with coronavirus people have become infected again after recovery? So the answer to this, I think, and I know about as much about this as you guys do through the news, is that coronavirus, I think, can come in several different forms. So some people are getting coronavirus and finding that they have quite a chesty cough and they have quite a few days at home, but then they're okay again, and other people have to be taken straight to hospital and put on a ventilator. So I think there are different forms of coronavirus. BCG is another vaccination. I can't remember exactly what BCG is for, but that's another vaccination that we get. used to get. Not sure whether we get BCG anymore. Why do many people refuse to vaccinate apart from having medical conditions? Some people can be affected by news that vaccines can produce other side effects. So there was a study that went around saying that vaccination was linked to autism. Since then, that has been disproved. That scientist was cheating. Vaccines don't lead to autism, but some people choose not to. Can you still get infected even if you've been vaccinated? Occasionally, yes, because there can be different forms of some disease. It doesn't happen very often, and it usually means that you get over it quicker. Oh, someone's helpfully put that BCG protects you from tuberculosis. That's right. I remember that now. Okay. If you've already had measles, is it necessary to have a vaccine? You've probably got some immunity, but it's probably still worth getting some medical advice for there, because it might be a different form. Okay, we're going to have to move on now. How can antibiotics help treat diseases? So antibiotics are substances that slow down or stop the growth of bacteria, curing disease. So they work to kill bacterial cells. They work to kill those alien cells in the body. An example is penicillin, which was discovered in 1928 by a guy called Alexander Fleming as a mold on bread. So that's what penicillin looks like there. Before it's been distilled down to its true form. So when you take penicillin, you're not eating mold, but you are eating something that was derived from mold. So antibiotics, work by damaging bacterial cells. Specific bacteria need to be treated by specific antibiotics. So we talked about the antigens, that are the proteins on the outside of pathogens or bacterial cells. So antibiotics are used to target specific pathogens. So a range of antibiotics is needed for a range of different diseases. What about viruses? Antibiotics cannot kill viral pathogens because viruses reproduce inside the host cells. So the host cells would be damaged too. Antiviral drugs are difficult to produce and viruses adapt quickly, so the drugs need to be changed regularly. We did talk about this last time, I think, when we talked about how viruses and bacterial pathogens differ. How do painkillers fit in? So painkillers are used to relieve the symptoms of a disease, but they don't kill the pathogens themselves. So for example, you would take paracetamol, which is a painkiller to treat a headache, but you've probably got a headache because of something different. So you might have a headache because you've got the flu, or there are lots of other diseases that can have the symptom of a headache. So a painkiller treats the symptom, but not the disease. Next, you need to learn about antibiotic resistance. So I've got this kind of flowchart here. Resistant bacteria to antibiotics means that antibiotics don't work. They cannot kill the pathogen that they're supposed to kill because the pathogen has mutated. So it's become different. So the antibiotic doesn't work on it anymore. So at the top, we have the normal situation. So bacteria grow and reproduce. And remember, bacteria reproduce really quickly. So you get lots of them very fast. Most of the time, the antibiotics kill almost all of the bacteria. You start to feel better. And after 1 week, all of the bacteria have been killed and you make a full recovery. You take all of your antibiotics, don't cut the dose short. That's what should happen. But if you add the antibiotic and by chance a bacteria has mutated, and doesn't respond to the antibiotic, even if the antibiotic kills all the others, that mutant bacteria can grow, reproduce in the presence of the antibiotic. Because all of the others have been killed, there's no competition, so they can reproduce and have all the resources that they need even more. And so the population rises of the resistant strains. You get lots of these bad bacteria that you can't kill with the antibiotic. So this is natural selection in bacteria, but because bacteria reproduce so fast, this can happen quite fast. So why has antibiotic resistance become a thing? Why have some bacteria become resistant? So firstly is overuse of antibiotics. So if patients go to the doctors and demand antibiotics for a viral disease, such as a cold, the antibiotics are wasted. They don't work on viruses, and colds are viruses. If you fail to complete a course, so often when patients feel well again, they stop taking their antibiotics early. They think, I feel fine now, I don't need the drugs anymore, I'll just carry on. That's fine, your immune system can probably cope, but it does leave some more bacteria alive for longer that can mutate and become resistant. And finally, in the past, antibiotics were used regularly in farming to keep animals healthy so that they grow faster and can feed us faster. However, if you're using antibiotics on every animal, say regularly, then it was thought that there might be some spread of antibiotic resistance from animals to human hosts. So now in farming, antibiotics are only used on the animals that really need them, which we hope would stop there being so much antibiotic resistance. Ways to prevent resistance, we've kind of talked about that already. So only take antibiotics when necessary, so when you have a bacterial infection. Use the specific antibiotic that you need for that pathogen. So don't take lots because you're not sure what you're infected by. It's better to get tested and make sure that you're taking the specific antibiotic. Keep hygiene levels high, then you won't run into the pathogen in the first place. And isolate patients with resistant bacteria. So if a patient in a hospital isn't responding to treatment, sometimes they'll be isolated. Hopefully you can contain that resistant bacteria from spreading to someone else. Okay, another mini quiz. So question 1, do antibiotics work on viruses? A, yes, B, no. Good, lots of B's. And question 2, which of these is not a way of preventing antibiotic resistance? A, good hygiene levels. B, using antibiotics regularly on all livestock on farms. C, finishing a course completely each time. Or D, isolating patients with resistant bacteria. Should have lots of B's for that as well. Good. Okay, any questions about antibiotics and antibiotic resistance? If you have a vaccination but your immune system isn't working as well, could you get the illness that you had a vaccination from? In some cases, yes, but I think those are quite rare. If you get a disease and recover, can you get the disease again? We did talk about that. So if you get a disease and then recover, you might get a slightly different form of the disease. That's why we get colds repeatedly. So we say, oh, I've got a cold. But actually the cold virus changes regularly. And so that's why we keep getting the cold, because it's a slightly different virus that we're getting each time. Will a virus kill humanity before the sun explodes? I have no idea. Okay. Last section. So we've talked about our first line of defense, so our physical and chemical barriers and our immune system. We've talked about how vaccines work and herd immunity, and we've talked about antibiotics and antibiotic resistance. So antibiotics are a type of drug, and you need to know about where we find the things that lead to drugs, and you need to know how we test drugs as well to make sure that they're safe. So that's what we're going to do now. So where do we find new drugs? So traditionally, we found new drugs in plants and microorganisms, so in our natural world. So for example, the heart drug called digitalis originates from foxgloves. Foxgloves are really poisonous, so don't go and eat them because they're good for your heart. That's not how this works. So foxgloves contain a compound from which scientists were able to extract digitalis and make it safe to use on heart drugs as part of heart drugs. The painkiller aspirin originates from willow. So we've got a picture here. Quite often animals will eat bits of willow, and that's because it does have some pain-killing properties. So you'll quite often find that sheep or cows, if they're turned out with some willow, they'll go and eat some of that when they're in a bit of pain. And then penicillin, which we talked about earlier, was discovered by Alexander Fleming from Penicillium mold, which is normally on bread. Most drugs are made in a laboratory by chemists in the pharmaceutical industry. That's the name of industry that produces drugs. However, the starting point may be a chemical extracted from a plant or a microorganism. So the origins of the drugs are usually in a plant or a microorganism, but then the drug themselves has been developed from whatever was in the organism and then made safe to use. So how do we know that drugs work? So there are several stages to developing and testing drugs. So we want to make sure that drugs are effective, that they're safe, and that they're stable. So we test drugs for dose, efficacy, and safety, side effects. So we want to know, does it actually work? So efficacy means whether it's effective. So does it actually work against the disease? We want to know what dose we need to use to do that safely. And we want to know if it's safe. Does it have any side effects? Toxicity, that's the word, rather than safety. Toxicity. So all drugs go through cycles of testing. So we have a disease, we've got a problem with a disease, coronavirus. We develop lots of different drugs and then we test them. That's what scientists are doing at the moment for coronavirus. So first of all, they do what's called preclinical testing, where they test on tissues and cells, and sometimes organs, and also on animals. So they do this to make sure that the drugs are safe and effective. So we haven't gone on to dose yet, but we want to know whether drugs do work against the disease and whether they have awful side effects that cause much worse problems than the disease itself. That's why we test medical drugs on animals normally. So cosmetics, we don't test on animals anymore, and anything else that isn't to do with health. So testing on animals is usually a last resort, but it does happen for medical drug development. After we've done the preclinical tests, You do tests on healthy humans. So you take volunteers for being part of the medical trial, you assign them randomly to groups, and then you test them with different doses of the disease. And you might also use something called begins with P. Mind has gone blank. A placebo. So a placebo is a pill that you might give to someone, or an injection that doesn't have the drug that you're testing in, but it makes the person feel like they might have had it. So for example, if you had two groups of volunteers and you wanted to test one with a certain amount of a drug in a pill, you might give the other group a placebo pill. So they still knew that they'd taken a pill, but they weren't sure whether it would make them better or not. So that eliminates people's ability to take a drug and then think. So some people take a drug and the act of taking the drug makes them feel better. So because they've taken a pill, they think, oh, I'll feel better now. And so they have a mental perception of how they feel. Whereas if you've taken the actual drug, that should be working within your body and you should be feeling better because it's combating the disease. So finally, you can test on people with the disease to check that it still works, and then find that optimum dose. And the final stage is publishing the results after peer review. This is really important. So peer review means that it's looked at by other people as well as just the scientist that's done the test. So it means that that science is looked at by other scientists and everyone concludes that it's right. So it just should prevent what happened with the link between autism and vaccines. It means that scientists are less likely to be able to cheat or to do bad science. Okay, another mini quiz and then we'll do some past paper questions. So, question 1, where did we traditionally find new drugs? A, in plants and microorganisms, or B, in laboratories? A, good, really good. And 2, which of these is not a thing that new drugs are tested for? A, safety, B, effectiveness, C, dosage, or D, looks? Good, lots of D's there. So drugs aren't tested for how they looked, but they are tested for whether they're safe, whether they're effective, and what dose we need to effectively combat the pathogen. Okay, any questions on testing before we move on to some past paper questions? How long does a vaccine usually take to be made? That's a really good question. So it depends on the vaccine, but all of that testing takes a long time and then you've got to publish the results. So the fastest would be about a year or a year and a half, probably, depending on how much you knew in the first place about what you could use for that vaccine. Good one there. What does it mean for a scientist to cheat? So a scientist can cheat by making up results. They can also cheat by what's called biasing the results of their experiment. So making an experiment, designing it so that it should lead to a particular answer. So rather than having, say, random groups, you could make sure that the people that you knew would be less affected by the disease were the ones that had the drug. So then it would look like the drug worked really well, whereas actually those people would just recover quicker anyway. Is a placebo used in a double-blind trial? Yes, exactly. That's exactly when it's used. So a double-blind trial means that a volunteer person within that trial doesn't know which drug that they're taking. So yes, they would have a placebo. If a drug works on an animal, does it always work on a human? That's a really good question. We usually test on animals that have quite similar systems to humans. So mice, because they're mammals, are often used. They have quite similar systems. For anything more advanced, sometimes you use animals that are much more similar, so it has been known to use chimpanzees. Can some people be allergic to penicillin? Yes, they can. So that's something to be aware of. Just like some people can be allergic to any sort of drug, I suppose. You can have an immune reaction to it. Okay, I'm just scrolling down. Chat. Okay. Someone's just asked what causes an allergic reaction. That's a really good question. So sometimes our bodies get confused, and it can be for all sorts of reasons. Sometimes it can be in our DNA, sometimes it can be things that we're exposed to, chemicals that we're exposed to, and that can affect our immune systems. So for example, if you have hay fever, I get hay fever, and it means that when I am exposed to certain types of pollen, I get a really runny nose. So my immune system goes, hey, we shouldn't see that, and produces loads of mucus, which is quite gross. Okay, let's do a past paper question, and then we can have some more questions as well. So if you think of anything else, do pop it in the chat, or the question and answer. Okay, so use the words from the box to complete the sentences about curing disease. So I'll show you the question now, and then I'm going to show you the worked answers on the next slide. So you don't need to worry about answering these unless you particularly want to. So when we're doing a past paper question, I always say underline the key words because it's really easy to misread them, particularly if you think they're going to be easy questions. So we've got 5 words. The substances made by white blood cells to kill pathogens are called. Substances made by white blood cells to counteract poisons produced by pathogens are called. Medicines which kill bacteria are called. And then we've got a part B as well. The MMR vaccine protects people against 3 diseases. Write down the names of 2 of these diseases. Oh, my plan was to write them for you. Okay, in that case, let's put them in now. We'll put them over here. Substances made by white blood cells to kill pathogens are called antibodies. And then the next one, right after here. Substances made by white blood cells to counteract poisons produced by pathogens are called antitoxins. Medicines which kill bacteria are called antibiotics. Remember, antibiotics only work on bacteria. And then we talked about the MMR vaccine, so you could have any 2 of measles, mumps, rubella. Okay, part C, now we have a table as well. let's underline words here. So all vaccinations involve some risk. Table shows the risk of developing harmful effects from the disease of a child is not given the MMR vaccine, and if a child is given the MMR vaccine. So the harmful effect they've listed on the left-hand side here. So convulsions, meningitis, or brain damage are all listed as possible side effects of the MMR vaccine. So the risk of developing the harmful effect from the disease. The harmful effect, this is why you should read the question thoroughly. From a disease if the child is not given the vaccine, or the risk of developing the effects if the child is given the vaccine. So the harmful effects of the disease, so of measles, mumps, or rubella, could be convulsions, meningitis, or brain damage. And the risk of those effects, if you're not given the MMR vaccine, is 1 in 200 for convulsions, 1 in 3,000 for meningitis, and 1 in 8,000 for brain damage. And the risk of developing those effects if you have had the vaccine is 1 in 1,000 for convulsions, less than 1 in 1,000,000 for meningitis, and 0 for brain damage. A mother is considering if she should have her child vaccinated with the MMR vaccine. Use information from the table, that is a key phrase there, to persuade the mother that she should have her child vaccinated. So this question is asking for your biological knowledge, and it's also asking you to read this table properly. And you need to show that you've read it properly by listing some sort of detail in your answer. It's 2 marks, so you don't need to do an awful lot, but you need to have 2 things that they can give you a mark for. So you've got less chance of getting all of the conditions if vaccinated. Okay, 1 mark, that's good. For example, you're 5 times less likely to get convulsions if you're vaccinated. So 1 in 200 without the vaccine and 1 in 1,000 with the vaccine. So that's a difference of 5 times. You could also say any of the others as well. So you could quote directly from the table saying that there's no chance of brain damage, but there was a 1 in 8,000 chance without being vaccinated. Okay. Let's have a few more questions, I think, just in case there's any that I've missed. And then we have got 1 more past paper question if we want to do that. Frederica, have you seen any questions that I've missed? There's been quite a lot on the chat, so I probably have missed some.
Yeah, I think there's quite a few questions, but I think actually you've been on top of them because quite a few of them are replicas of each other.
Oh, good. Okay.
So I think it's probably best to move on to doing the last past paper question. And if anyone does have any more questions, then you've just got a couple of minutes to get them through now.
Okay, brilliant. I have seen one person actually just quickly saying that they're in year 11, but they're revising for GCSEs and they're going to go on and do biology for A-level. Is there anything you can do to prepare? Yes. Make sure that you've got your GCSE content down. There's loads of topics that directly translate over to A-level. And there's lots of sites where you can find revision materials for A-level as well. So one that I quite like is Physics and Maths Tutor. That one's quite good. Got some good revision materials. Okay, question 5 here. Microorganisms cause infections when they're acting as pathogens. The human body has many ways of defending itself against microorganisms. Describe 2 ways the body prevents the entry of microorganisms. Okay, so we're talking about that first line of defense here. So you need 2, but we talked about quite a lot. So you can have acid in the stomach, kills food in pathogens. Pathogens in food, that's supposed to say, not food in pathogens. Skin forms a barrier against pathogens. hairs in the nose help to trap pathogens. And then we talked about how the trachea has mucus and also little hairs that trap pathogens, and then the cilia waft the mucus to the throat to be swallowed. Because those are connected, you have to either say both, or you could have one or the other, but you couldn't have them as two separate points. So you need to talk about what happens in the trachea and bronchi and what happens somewhere else as well. You could talk about tears there as well. You could talk about the antibacterial enzymes in saliva that we talked about too. So there's plenty of opportunities there. Okay, and that was the last question that I had there. Our next session is going to be on exam technique in this particular topic. So if you're looking for more guidance on how to answer questions that come from this topic, we're going to talk a lot more about there. So we'll talk a lot more about vaccine questions. We'll talk about how to answer questions with graphs. We're going to talk about how to answer those long essay questions that you sometimes get for 6 marks. So that will all be next time. Okay, so I think we'll just spend the last couple of minutes answering more questions. Got someone who really wants to know if using the CGP revision guides for science is fine. CGP revision guides are really, really good, yes. You probably want to combine them with your own revision as well, so making your own. It depends how you learn best, so people learn differently. People can learn best by seeing things or hearing things or doing things. So it's best to do a combination of all three until you know what works best for you. So make mind maps, stick post-it notes everywhere, write out everything you know about a subject and then see how that compares to the specification. And I'd highly recommend as well going online and getting a copy of the specification. You can if you Google it. And then you can have a look at what they say that you need to know. It doesn't include absolutely everything down to the last detail, but it does include an idea of what you need to know. Okay, scrolling through, trying to see whether there's any more. Do tears come only when there is bacteria because our eyes don't constantly tear? Well, actually, that's quite interesting. So our eyes are never dry. Our eyes always have a layer of kind of liquid in there, not necessarily tears, but they do always have that thin layer of liquid on there, and that contains some antibacterial enzymes or anti-pathogen enzymes as well. So it's not always just bacteria. But if you're going along, because this happened to me the other day, and you get a fly in your eye. I was running and there was a fly in my eye. And you automatically cry, don't you? And you cry because your eye is trying to get it out. It knows it shouldn't be there. And a fly isn't necessarily a pathogen, but I bet it's carrying stuff that could lead to some nasty things if it went behind my eye and all the way into my body. So the tears are a way of stopping that happening. Why do you produce tears when you cry from emotion? Well, that's something different, isn't it? So that's to do, your emotions are linked to your brain, and your brain linked to your nervous system, and your nervous system is linked to all of the different parts of your body. So you cry because your brain is triggering that response. I don't know much more about it than that, actually. There's probably a bit more behind it. If people can't produce tears, is there an alternative for prevention near the eyes? If people can't produce tears, I imagine they're given some sort of eye drop to use regularly to keep the eye in good condition, if nothing else. And without tears themselves, like I say, that layer of liquid on the eye, which can become tears, is there. Is it possible for pathogens in your stomach to survive? Very occasionally, yes, but only very occasionally. So right at the beginning, when we first started doing these online school sessions, so you may have been in that session or you may not, we talked about digestion and how different enzymes and different proteins and different microorganisms have optimal pHs. We talked about it particularly for enzymes, but some have optimal pHs. And so it's very rare that a pathogen can tolerate that really low pH in the stomach. Very occasionally they might be able to. Okay, I think we're going to have to leave it there, I imagine.
Yes, I agree. I think that's all we have time for today, everyone. But thank you so much for your questions, and thank you for joining us for this session. And look out on the My Tutor online school timetable for the rest of the sessions for this week. Thank you, Charlotte. Thank you, everybody. Bye.
Bye, everyone.